last period to discuss about the structure of synapse, types of synapse. Then about transmission of impulse. Before that, we will discuss few terms. Neurons are the types of nervous tissue, nervous cells from nervous tissue. Those are participating in the process of conduction or the electrochemical or bioelectrical disturbances in the form of impulse. Impulse means the bioelectrical or electrochemical disturbances or change from the surrounding, and due to which that nerve cell becomes excited. Excitation means that due to this impulse, the cell's membrane are charged. That means nerve cell membranes are charged. These charges are carried from axon terminal, from body of the cell to the axon terminal. So this charged membrane helps to conduct that impulse from site of excitation, site of stimulation to the axon terminal. The membranes of nerve fibers consist of voltage difference. This voltage difference which is created due to the difference in the material present inside the cell, axon fiber, and outside the cell, extracellular fluid and inside the membrane. Mostly substances and the amount which is roundabout equal. That means number of ions are equal. But there is a slight difference. Outside the cell nerve fiber, number of sodium ions which is more. Inside the cell axon fiber, number of potassium ions which is more. At the same time, inside the cell there is a presence of negatively charged molecules like amino acids, negatively charged molecules like proteins, due to which inside the cell or cell cytoplasm which is slightly electronegative, while extracellular fluid which consists of sodium ions in a large quantity, so ECF which is slightly electropositive. We know that when this condition appears, that means sodium ions are moving outside, potassium ions in cell cytoplasm with negatively charged proteins, negatively charged nucleic acids, the voltage is created. That a potential difference which is about -17 millivolts. But this potential difference which is restored with the help of the sodium and potassium pump. That means actively again the concentration gradient, again the electrical potential gradient outside the cell cytoplasm, amount of sodium ions which is maintained in more number concentration. Inside the cell potassium ions concentration is maintained using energy, using ATP molecules. This process maintains or restores this potential difference inside the cell and outside the cell. That means on the both sides of cell membrane. This condition which is maintained with the help of these potassium and sodium ion pumps, and these both pumps are voltage gated channels through which these pumps are operating. So this condition which is maintained forcefully, that condition which is observed during resting stage. That means when that nerve is not working, that time the situation is observed. Means is a resting axon. Inside which there is negative charge, outside which there is positive charge. The sodium ions are maintained outside, potassium ions are maintained inside. With this potassium ions, this nucleic acid and proteins maintains electronegative condition inside. Due to the presence of large quantity or number of potassium ions, electropositive condition is mentioned outside, and between these two there is electropotential difference which is about -70 mV which is maintained, and this potential difference which is at resting state of the axon, so called as resting potential. Then what happens when there is a generation of nerve impulse? Impulse when it is created, actually impulse which is created due to the electrochemical or biochemical disturbances from the surrounding. Due to this any change, what happens? There is influx of sodium ions. Sodium ions enters inside. Due to which, what happens? This resting potential or potential difference which is minimized. Actually, this resting potential which lies between -50 to 100 MV, round about -70 MV. But here, due to any change, due to the entry of sodium ions, the potential difference which is minimized, and due to which rapid influx of sodium ions takes place. That time potassium pump which is not working. So inside the cell, sodium ions enters. Due to this entry of sodium ions, positively charged sodium ions, the part which is depolarized and axon is excited, where inner fluid or cytoplasm of the axon fiber which becomes electropositive, and outside this axon fiber which is become electronegative. This is due to the changes in the electropotential difference, and which lies at about +30 to -60 MV. In this way, here electropotential changes are observed, and that axon portion becomes depolarized. Due to which another next part where also action potential, this action potential stimulates, and that region becomes depolarized. That means this depolarized portion pulled in forward direction due to this potassium difference, due to the working of sodium and potassium ions. So excited condition, impulse is created in this manner due to the depolarized condition. This depolarized condition due to the changes in the electropotential difference means what happens during resting condition, electropotential difference which is about -60 MV. Outside the cell there is electropositive, and inside the cell which is electronegative. But due to the any change, disturbances, sodium ions enter, sodium pump activates, sodium ions enters, and inner material cytoplasm becomes positively charged, depolarized, and potassium ion goes outside. Due to which a stimulation is created, and which is carried forward direction again by formation of this depolarized portion of the axon fiber. Then repolarization means after depolarization the portion becomes repolarized. What happens when influx of sodium ions on the large scale. Gradually, this entry of sodium ions gradually slows. At the same time, potassium pump is activated. In large quantity, potassium ions are given out, and that's why the sodium ion pump slow and ultimately stops. But potassium ion remains active. That's why charges are changed. That means this sodium ion entry which is stopped and this potassium ions are actively given out, due to which again changes in the electropotential or electrical potential takes place. Then negatively charged, that portion becomes and positive charge outside takes place. That means this structure goes in forward direction. That means resting potassium to the depolarization, depolarization to repolarization. Again, this type of changes are observed from one node to the another node of body wire in the medullary nerve fibers. That means wave of carrying impulse which is not simply wave of electropotential difference, but where the changes are observed due to these transport pumps and gated channels only. The time required to change in the polarity, that means from polarization to depolarization, from depolarization to repolarization, means polarized changes in the depolarization to repolarization requires a short period which is called a refractory period. Here, transport of that impulse in the form of electrical potential changes and which is transported from one node to the another node. That means jumping, not carried like a wave, but jumps from one node to another. Why? Where transport pumps and gated channels are absent, where myelinated sheath is present. These transport pumps and gate channels are only present in the nodes of blood wire, and where only the electrical potential differences are found, and from that which is observed in the another node. That's why these charges are jumping from one node to the another node. This condition is called as saltatory conduction. Saltatory means jump from one node to the another node. Here in the nodal portion, what happens when there is disturbance, sodium ions enters, due to which there is a depolarization. Then sodium ion stops entering, potassium ion goes outside. The outflux increases, due to which there is a repolarization. Then depolarized part goes in forward direction. Again, the repolarization takes place in this way. From one node, this condition takes place. Then there is no transport from my gated channels in this medullary nerve fibers where Schwann cells are present, only present where node is present. So from one node to the another node, there is jump of this charge. This is about how the transmission of impulse takes place. So this process takes place in the nervous system of the human being. Human nervous system consists of three parts: central nervous system, peripheral nervous system, and autonomous nervous system. This nervous system is centrally situated on the axis of the body. That's why called as central nervous system. From this central nervous system arises many fibers. Those are carrying or transporting impulses, messages to the related organs with the help of nerve fibers. Those are peripherally situated but arises or connects to the CNS, cerebral nervous system, and this nervous system autonomously working, no doubt, which is also connected to the CNS. So we will discuss one by one. Our CNS, central nervous system. In case of human beings, central nervous system is very well developed. The central nervous system consists of two parts: brain and spinal cord. The brain, which is situated into the cranium or belong into the cranium. Cranium is the brain box. The spinal cord, which is nothing but a terminal end of the brain. We will discuss afterwards. That the spinal cord, which is protected by the vertebral column, that is bony covering. Inside the brain and inside the vertebral column, there are three meanings or coverings. These meanings are protective in function. These meanings are of three types. The meanings, first meaning that a durameter which is close to the bony box, cranium, or very close to the vertebral column, which is nothing but durameter, which is formed from fibroelastic material, which is protective in function, which is tough in nature. Below this durameter there is a space. Below this durameter there is a space called as subdural space. This subdural space, which is filled by shear fluid, which is protecting in function. Below this durameter there is a arachnoid matter, which is spider web like vascular delicate part. This arachnoid matter, which is very important. Below this arachnoid matter there is a space which is called as subarachnoid space. Below this arachnoid matter, this subarachnoid space, which is very important, this subarachnoid space which is filled by CSF. The coronal plexus present in the arachnoid matter or in these meanings also see the CSF, so important. And inside, which is very close to the brain, upper layer of the brain, which is a thin, delicate vascular pie. This pie matter which is nourishing in function. So all are important, and inside which brain is present. Just before this, we have discussed about CSF, cerebrospinal fluid, which is lymph-like colorless fluid, which is present in the cavities present in the brain. Our brain, which consists of cavities, brain cavities called as ventricles. Ventricles are filled by CSF, as well as which is also present in the central canal, CSF, which is very important. The CSF, which is secreted by the arachnoid space as well as ependymal cells. The CSF, which supplies adequate amount of water and avoid desiccation of the brain cells. The CSF carries food material, nutritious material, and is supplied to the nerve cells. The CSF collects waste material, nitrogenous waste material produced during the metabolic activities of the nerve cells. The CSF, which act as mechanical shock absorber and absorb mechanical shocks and protects our brain. So this CSF, which is very important. In the medulla oblongata region, there are three openings by which CSF released into the blood, and the CSF is uniform. Its special gravity, which is about 1.005, and its total amount is about 100 to 120 cc. So this CSF very important. So we will discuss about afterwards about brain and